you have really stirred the pot this year with one of the most popular and most talked about science papers of the year, with hundreds of media mentions and controversy across the biomedical world, with a paper landing that claims to demonstrate that the heritability of lifespan is approximately twice as high as any study has previously indicated.
The previous estimates for lifespan heritability were based on cohorts born in the late 19th century. And just to put it really simply, the world back then was very different than today. People died at young ages, in the age where infectious diseases were rampant and antibiotics weren't available. And dying at 35 from tuberculosis is not aging. We're interested in understanding how heritable is lifespan when the majority of us reach age 70, 80, like in modern society. That's the main message. And we do come up with an estimate that is approximately twice as high.
Welcome back to the Sapiens Podcast. I am delighted to be welcoming Ben Shenhar, a doctoral candidate from the Weizmann Institute of Science. And you have really stirred the pot this year with one of the most popular and most talked about science papers of the year, with hundreds of media mentions and controversy across the biomedical world, with a paper landing that claims to demonstrate that the heritability of lifespan is approximately twice as high as any study has previously indicated. Welcome to the podcast, and break this down for us.
Okay, thanks so much for having me. Yeah, I would say the previous estimates for lifespan heritability were based on cohorts born in the late 19th century and even before. And just to put it really simply, the world back then was very different than today. People died at young ages from causes that aren't, let's say, directly related to biological aging. Dying from pneumonia or tuberculosis in the age where infectious diseases were rampant and antibiotics weren't available. And the original estimates were based on those cohorts, these historical cohorts. And what happened, I think, is that people, took the estimates from these studies being informative to the genetics of aging. And what we claim is that those studies weren't actually examining aging. Dying at 35 from tuberculosis is not aging. We're interested in understanding how heritable is lifespan when the majority of us reach age 70, 80. Like in modern society. And that, I think, that's the point of the paper. That's the main message. And we do come up with an estimate that is approximately twice as high, around 50%.
So for folks who are not really used to what heritability estimates are describing, can you give us a better understanding of what it means when we assign a percentage of heritability to a particular trait or outcome?
Sure, yeah. So heritability, I would say, is a notoriously tricky concept in that it's very easy to talk about heritability, but I think often people get confused about what it actually is. So heritability is not saying how much something is defined by your genes. It's saying how much of the differences between people is due to genes. And the difference is that the value itself of any trait, if we're lifespan or we're talking about height or whatever, always depends on the environment. So we can't just say the genetics are 80% important, 80% define one's lifespan. But what we can say is when we look at a group of people and we see that there's some difference or variance in scientific terminology between their lifespans or between their trait that we're looking at, we want to ask how much of that difference is due to genetics. And that will always depend on environment. So just to, let's say, for example, if we were to take a society and have everyone, live really poorly, everyone eat, fast food all day and smoke and drink, heritability would go up, not down. It would go up because the environment just became homogeneous. It means the environment became the same for everyone. So there's lots of pitfalls, I think, in terms of sometimes people get confused about what it means. And of course, it depends on environment. So in that sense, we're not saying that previous estimates were wrong, previous studies, because the previous studies examined heritability in a different environment, in the environment of the 19th century and even before. And what we're claiming is that that environment is not what we're interested in when we talk about aging, the genetics of aging, because of the fact that lots of people were dying, before reaching old age. So heritability is talking about the differences in differences between people. And we're asking, in an ideal or idealized kind of environment where we don't have lots of external risks and hazards to our life, when people reach 70, 80 years old, how much of the difference is then between when people die is due to their genetics? That's what we try to answer.
There's a really common remark that you see floating around when we talk about how long humans live that says something to the effect that, wow, we've doubled our lifespan in the past couple hundred years with the advent of the modern world. For some people, they're attributing it to the trappings of the safe modern environment. For some people, they're attributing it to modern medicine. But in either case, the reduction in these sort of external causes of death driving humanity from a situation where we're living in our 30s on average to where we're now living in our 70s or 80s on average. Can you describe for us this important difference between what this sort of average life expectancy is and what we mean when we talk about the actual maximal lifespan for humans?
Yeah, sure. I think that's a super important point. And you're right. I mean, over the past 150 years, life expectancy has gone up from around, 40, 50 to around 80 plus in developed countries. And that was driven by several factors, I would argue none of which target actual aging. The first one is a reduction in child and infant mortality. It's quite cool. You can see that the chance to die in childhood has been decreasing exponentially since the year 1900. So that's the first thing. The second thing is extrinsic mortality, what we talk about in the paper, which is kind of the risk of dying from non-biological aging, which is due to prevalence of infections, accidents, violence, and so on. Just to get I guess we'll talk about this in more detail shortly, but just to get a feel of this. So the chance to die in 1880 from age 20 to 40 was 1 in 5. Okay, that's crazy. Like you reached age 20, by the time you get to 40, 1 out of 5 of your friends, unfortunately, has died. Nowadays, that would be a tragedy. But back then it was much more common, and it's not because those people aged. So, they were 30-year-olds who had gray hair and were like the 90-year-olds of today. No, there's all these external hazards that unfortunately were killing people. And finally, I would say this is something that we're researching right now in our lab. You see that actually it's very interesting. So I would say in the late 19th century, beginning 20th century, the majority of longevity gains were from childhood, and then from young adulthood, which is this removal of extrinsic mortality. Then around the 70s, we see the cardiovascular revolution in a way. We see the introduction of statins and lots of bypass surgeries, lots of new medical interventions, which are actually pushing up lifespans on the other end. On the end of so just to give you like a concrete example, my grandparents, both of them died from heart issues, quite young, 50, 60. Today, that probably wouldn't have happened. Today, they would probably have been treated, and I would assume that their lifespan would have been, 80 plus. And we really see that happening around 1970. So again, that's also not about slowing down aging. It's about, I would say, mitigating the deleteriousness of aging. In other words, making us more robust to the harms of aging. But we're still aging at the same rate. That's what I would argue. So just to sum up all that answer, in the beginning, it was child mortality that was pushed down. Then it was kind of external hazard that was pushed down. And then in the late 20th century, it was actually the old age mortality, which was pushed down due to medical innovations and medical interventions.
And I think that's a really important distinction to make, because the community as a whole, the world and investors, have a burgeoning interest in this idea that we have longevity medicine, longevity biotechnology, that in the same way as I've heard it framed a number of times now, in the same way that we have pushed life expectancy from, say, 40 years old to 80 years old, why not be able to push it from 80 to 120 or even beyond? And of course, there are some advocates of these longevity interventions who are claiming we could even push it past then. And I think the really important part of what you just pointed out there is that the majority of the average life expectancy moving from the 40s to the 80s has been the reduction in the first part of that bimodal mortality curve, that we've reduced the loss of life early in life. And only in recent times have we started to actually address some of the diseases associated with aging. And so the question becomes, how much of that can we split off into aging that is derived from things going on inside of the body and from things that are still going on outside of the body? Whenever I'm interacting with a patient, I'm always having a conversation both about what we can do to care for what's going on inside of their body, and of course, what lifestyle components are contributing perhaps towards early mortality, like the onset of early cardiovascular disease. And there's a really high curiosity that a couple of your papers have touched on about whether lifestyle versus heritable factors are actually the determinant of lifespan. How does your paper move the goalposts on how we understand that to be, and how did you arrive at those results?
So regarding the heritability, the genetics, so we, in our lab, we're quantitative people. So I myself come from a physics background. I've been in the aging field for 2 years. Okay, that's it. I had no prior training in biology or whatever. My master's was in theoretical astrophysics. So I made a radical switch. So what we do in our lab is we try to physics aging processes mathematically. We try to understand, like, what physicists like to do is to take some complicated quantitative behavior and try to find what is the simplest mathematical explanation that captures its behavior, not in the sense of reproducing the statistics, but what is the mechanism involved. Right. So mechanism is very important to physicists. So we model stuff in our lab, and I was playing around with the model that we use often in our lab to capture aging, which maybe we'll talk about later. It's called the saturating removal model, and it conceptualizes aging as a balance between damage that is accumulated and removed. And you ask how that damage changes over time. And you model death or disease when damage crosses a particular threshold. I want to point out that general framework is not unique just to the SR model. Recently, there was a preprint out by Fedichev and Gruber, who have their own minimal model, which is very similar in many respects to the SR model. So this kind of conceptualization of aging as, something being removed, produced and removed, the way to picture it pictorially, I think, is very easy to think about as think of it like a particle or a ball in a well or a valley. And when the ball escapes from the valley, you die. And aging is the valley becoming flatter and flatter. That is aging in terms of the model, in terms of the mathematics. So imagine a ball stuck in a valley and the mountains are getting flatter and flatter until there's no valley anymore. There's just a plain. Okay, so using this kind of framework, we were interested in understanding what happens when you introduce heterogeneity or changes between parameters into people, because people are different in how they age. And we were trying to calibrate on twins, and then we kind of found out that I found out that when I try to do my calibrations, there's a very important aspect that I have to include in my calibrations, which is the amount of extrinsic mortality or mortality that isn't related directly to the aging process. So when we say that the ball that escapes a well dies, that to me is biological death, biological aging. But like we said previously, if you look at mortality curves or mortality, you would see that mortality curves, there's a plateau, a constant mortality from around age 20 to 40. And like we said previously, that thing's been going down for it's been going down tenfold in 100 years. And to us, that's extrinsic mortality. This is mortality that isn't directly related to aging. It's also constant, right? Aging gets worse. It's something that's dynamic. If it's a plateau, we say that it's extrinsic. And I noticed that that value of the plateau really, really changes my calibrations. Like the model is super sensitive to how much of that plateau I put in. And then I realized when we were calibrating and try to simulate twins using our method, that when we remove the plateau altogether, suddenly the numbers go up to around 50%. And that's where we kind of thought, maybe there's something going on here that led us to further investigate. And in the end, what we find, and I would say what we claim is that, again, it's around this 50%, 50% of heritability, 50% heritability of lifespan from biological aging. And I want to emphasize a crucial point. We also analyze a new data set that wasn't previously analyzed. Thanks to great collaboration that we have with Sarah Haig's lab from Karolinska Institute in Sweden, we had access to a SATSA twin cohort, twins raised together and apart from Sweden. And for the first time, we analyzed twins that were born in the 1920s to 1930s. So this is the oldest twin cohort that's been analyzed, because you need a cohort where everyone's died already in order to do this kind of research, right? That's why all these cohorts were born 100 years ago. And there we found that for twins born from 1920 to 1935, no model, no nothing, no extrinsic, intrinsic mortality, whatever, the heritability that we got was 44%. Okay. So we claim, based on our models, our modeling, that it's around 55%. On a cohort of twins born in Sweden, the majority of them died around age 80, which means they were dying around the year 2000 in the modern Swedish healthcare system. The heritability was around 44%. And that's model independent, and that has nothing to do with this kind of partitioning into extrinsic and intrinsic. So I think the main claim that I want to make is that, like I said, heritability is always dependent on environment. And in the environment of modern society, where people reached old age, where people reach the age of 70, 80, 90, the heritability that we expect of lifespan under those environmental conditions is around 50%. That's what we find. And the remaining, what you said, lifestyle, that's the remaining, that's the other pile, that's the other 50%. And there's a very important question, which you alluded to, which is, okay, so we have 50% that we think is genes. What about the other 50%? How is that split up? Is that lifestyle? Is that, diet, exercise? Is that noise? Is that randomness? So that's actually a new research that we want to do. That's like a follow-up kind of research we think is super interesting. My gut, by the way, I don't know, I may be wrong. My gut says that it's actually around 50-50. That means 25% lifestyle and 25%, for lack of a better word, I would say stochasticity or randomness. We know there's a lot of randomness involved in aging, right? There's just a lot of stuff that we don't really grow genetically identical organisms in identical conditions, and you still get like 20-30% variance in their lifespans, right? So of course there's some randomness here. We can't actually control everything. And so my gut says that actually lifestyle maybe would be only 25%. I don't know. That's a gut feeling. I haven't done the research yet.
That's a really interesting distinction that you make there too, because I think for a lot of people, there's a perception that there's almost essentially a false dichotomy, that it's either genetics or it's lifestyle. And it's really important to remember that in systems that develop with path dependency, systems that are inherently contingent, in the same way that in physics, we now know that the macroscopic structure of things like galaxies and galaxy clusters actually follow due to path contingency from tiny microscopic stochastic variations in initial background temperatures of the early universe. The same sort of thing happens because organisms that grow through cell division do ultimately inherit stochastic amplification of early minor variations. So something as simple as how a cell divides in a blastocyst can ultimately change the way that tissue differentiates 30, 40 years later. What the probability of something like a amplified somatic mutation can drive differences in the onset of cancer. And these things are not necessarily part of the nuclear genome. They're not necessarily controllable by lifestyle factors, but they can actually amplify the distinction in the lifespan between those two organisms.
I definitely agree. That's a great point. And so we did this division into genetics, lifestyle, and now we're saying stochasticity. Now I would claim that within stochasticity, we have a further division. And that further division, one half of it is what you said. That's developmental stochasticity. But there's also stochasticity, which is dynamic stochasticity, which means the randomness just from life. So that's another interesting question. So let's say that we think that 20-30% is stochastic or random. How much of that is developmental randomness, like you said? It certainly plays a role. I don't know yet how much. And how much of that is dynamic randomness? So I think that's a super interesting point. That, by the way, is true about, any trait that we're talking about. Like when we talk about heritability and stuff, like, a great example is personality. So twin studies find that, lots of personality traits are 50% heritable. I have a young son, and even before he could talk, he has a core personality. And to me, that is developmental randomness. There's something in probably his neurogenesis or maybe the hormonal environment of his womb. I don't know. But there's also another randomness that goes on throughout life.
Organisms have a deep ancestral contingency, which relates to the ancestral line that produces an organism. So something that resolved the differential survival and reproduction of your great-great-great-great-grandfather is a type of built-in path dependency, but it tends to be shared between siblings. And then you have developmental contingency, which once twins are divided, they're no longer down the same track. So they have different developmental contingency. And then, of course, you have environmental contingency. So something like the differential exposure to hormone environment, to toxins, to co-regulators of the genome, like the microbiome, all potentially can contribute. The question that our patients always have is always going to resolve around what they can do to change their lifespan. And work that you've been a part of has specifically made the claim that lifespan has a very limited potential impact from lifestyle factors. Can you tell us what you mean by that? And how should patients take the science for those of us, because all of us ultimately are patients at some point? What can we do? Is it actually worth eating kale salad and getting up at 6 a.m. and running and denying ourselves processed foods and all of the things that we constantly talk about as drivers of lifespan?
Yeah, it's a big question. Okay, so I have several things to say about that. So firstly, I would say I have no competing interests here. Okay, I don't have a company or whatever. So I'm not trying to sell anything, which is a big difference because a lot of people in the longevity business are trying to sell stuff like, academia, industry, whatever. So based on my research, I would argue that the amount of influence that we have first of all, to extend our maximal lifespan, I don't think that anyone can do that. I think we're all kind of capped at around, 115, 120. I don't think any of us can push to 130. So that I would get out of the way. Okay. I currently, based on what we know right now, current medicine and extrapolating what's been done. That's my opinion. We can get into that, why that's the case. Secondly, regarding not maximal, but let's say your, expected lifespan. So based on what we think, I would say that you can push it, plus minus 6-7 years in either direction. But I would also say that now this is kind of getting maybe a bit more philosophical. There's a great Yiddish author, Yitzhak Bashevis Zinger, Nobel Prize winning author. And they asked him in an interview once, Mr. Zinger, do you believe in free will? And he said, of course, I have no choice. Now, I love that. And what the reason I love that is because, okay, maybe you don't have all this control over how long, I don't think you can make sure you're going to live to be 140. But I think it's healthy to live your life as if you do have that control. I think if we give ourselves agency in the way that we live our lives, believing that we have control, even though it may not be grounded in science, I think that leads to more happier and a more empowering life. And also, I would say that, it's not only about, the amount of days you're on Earth. There's this other quote there, I think it's Lincoln, who said, it's not the amount of days in your life, it's the life in your days, or something like that. That's so true. I mean, so I mean, so if we think about this in terms of longevity, we're talking about health span. So what matters is your health span and your life satisfaction. So if you get up in the morning and you eat well and you exercise and you live a healthy lifestyle, I guarantee you your personal well-being will be much better than if you do none of those things. So maybe it won't push you to 140. I don't think so. But I do think it will make you live a happier life. And I think that should, I think much of the focus in longevity is how do we extend our lives. But I think a lot of the focus, which is, kind of the old focus of, classic philosophy, how do we live good lives? And I think all this healthy lifestyle is about living a good life. And that's great. And even if we don't have the control that, we would hope for.
I think there's a valid pushback to a lot of the longevity discussions that center around that idea that you're talking about, that we could focus perhaps more on health span, like maintaining functional resilience, maintaining strength and mobility and immune resilience, and the kinds of things that allow us to live a life that is sort of unencumbered by the end of life care that is a lot of the late life in many Western countries. And absolutely, the science seems to support the idea that the lifestyle factors can extend that health span, that we can live high functioning lives for longer percentages of our lives. The question of why we can't use that to extend will continue to capture the imagination of so many of the researchers in this space. And so the question I have for you is, why is it that if it is even 20%, if it's 25%, if it's even some double-digit percentage that is resolved by lifestyle differences, why is it the case that we can't add more years? Why is it the case? And this is a conversation that we've had with authors like Vedichev in previous seasons, but why do you think we're not able to push that sort of hard cap of 120? Let's just say we all do everything right and we kind of are able to mitigate those late life trade-offs like with cardiovascular disease. Why is that cap there?
Yeah, that's a great question. So regarding the how much influence we have, the influence is about, like, the question you asked, I think kind of combined two different aspects. So I'm going to pick them apart. One of them was about how much of your lifespan, let's say our median or expected lifespan, can we impact, and the other was about pushing the boundary, the maximal. So regarding, I think you were talking about the maximal. That's the why can't we push that?
And for the audience, let me kind of clarify there, because that's an important distinction in the mathematics that we don't. In many of the longevity discussions, I feel like there is a blurring of those two things. And that's a very important distinction making. What we can talk about life expectancy of an individual, we can talk about average lifespan of a population. When we talk in biology about a species maximal lifespan, what we are talking about is what the sort of upper limit of that adult lifespan is. So in something like sea turtles, their average life expectancy is very, very short, on the order of a couple of years or something, because the majority of them die very young. But if they survive to adulthood, they can live decades and decades and decades. With humans, the maximal species lifespan, as you were mentioning, is still kind of locked in to 110 to 120s range, and we don't have any evidence that that's ever been exceeded or can be exceeded. And why is that?
Yeah. So yeah, exactly. So I have a preprint from our lab, my work precisely on that, on this maximal lifespan thing. Like, why is maximal lifespan constrained? By what is it constrained, and why? So why is a very good question, and it's always a hard question. But what we found in our research, what I found in my research, is that if we go back to this picture of a little ball stuck in a valley, and when you jump out of the valley, you're dead, and it has noise. So the ball is kind of, jiggled around in this valley until, it's eventually flung out. And what we find is that, like I said, aging is the flattening of the valley. It's the flattening of the valley into a plane. And what we find is that the rate of flattening, which we call aging, mathematically speaking, is actually remarkably constrained among people. So notwithstanding aging pathologies, where that's where we think that actually flattens faster. But among, let's say, normal aging, even if, you look young or you look old or you're in good health or you're in bad health, actually we think that the valleys, if we will, of people of similar age are flat to a similar degree. And I think it's interesting because in other organisms, it doesn't seem to be the case. It seems that in other organisms, this thing is a lot more plastic. So it seems that in mice or in flies or in worms, you can change the flattening of their well. If they have a well, that's a kind of a different topic, but it does seem that the fundamental aging rates in other organisms are plastic to a particular degree. I would argue that also the longer an organism lives, the less plastic the aging rate is. So, worms who live the fewest, you can do a radical life extension. And flies, it would be less. In mice, it would be less than that. I don't know, 10, 15, 20% maybe, rapamycin, caloric restriction. In humans, if you do that extrapolation, you end up with like 2% or 3%, which is kind of what we argue in our paper from completely different reasons. That how much the flattening can change between people is around 3%. So I would argue that we seem to be optimized in a way. It seems that evolution somehow optimized our aging rates. We're at the end of some kind of optimization process. I'm not entirely sure why. But the fact of the matter is I believe that it's quite hard to push that. And I'm not saying it's impossible because, science and technology is a wonder and, humanity, the next frontier is always there. But I would argue that it definitely has not been done up to now. I have seen zero evidence that anyone today, any longevity company, any paper, any lab has any real method of extending maximal human lifespan. I myself have not seen evidence of that. And anyone who claims otherwise, I think, are being disingenuous. Maybe it'll happen. I'm not saying it won't happen, but I haven't seen it. I haven't seen evidence that it'll work today.
That is worth repeating again and again and again, because there are entire movements that claim to be founded on studies. There are, we'll say, actors in the longevity space who constantly repeat that notion that their interventions, whether they are supplements or whether they are lifestyle choices like calorie restriction or whether they're direct interventions like rapamycin, there are actors who continue to suggest that these kinds of choices and interventions are supported by studies that demonstrate lifespan extension. And inevitably, when they're based on lifespan variations in shorter lifespan organisms like mice that have this high plasticity, they're not translatable to humans. And this is something that bears repeating, but it's also something that evolutionarily is not surprising, because when you have an organism that only lives a couple of years in the wild, their presence of something like a couple of months of famine, a couple months of calorie restriction can impact the net reproductive output of that organism substantially. It can dramatically change the probability of passing those genes on. And so it makes a meaningful evolutionary difference, selectable by natural selection, to extend the lifespan of that organism by double-digit percentages, because this dramatically increases the chance that that organism can go through another reproductive cycle. And so this, we would expect to be under selection from natural selection, that there should be a sort of environmental response built in to things like calorie restriction. In humans, our long life isn't through the extension of our reproductive window. The longevity of our species actually has almost nothing to do with extension of the reproductive window, because we have the same reproductive window as other great apes that we're closely related to. What we have is a grandparent, a post-reproductive phase of life. And so calorie restriction, changing your late post-reproductive life by a few years, does not significantly impact the probability of your genes being passed on. So it's an underappreciated evolutionary prediction that we should not have high plasticity in response to things like a year, even a 2 year, even reduced calorie daily intake, because our extended lifespan is not during the reproductive window. And so it's not actually selectable in a significant way by natural selection, because you've already had your kids. You are no longer under selection at that point.
Right. Yeah, those are great points. I would say several things. So I would even go further and argue that even if you were to live on a caloric restriction throughout your entire life, I don't think it would do a meaningful maximal lifespan extension. That's what I would argue. And what you said is definitely true. And in that sense, when we talk about the heritability of lifespan, so I would say that, these are traits that are in the shadow of selection. Because when we talk about the heritability of intrinsic lifespan in our paper, we're talking about, again, lifespans which cluster around age 80. And, that is definitely natural selection. I don't think is working there anymore, right? We're talking about people who have already reproduced, probably their children have reproduced. You could do the grandmother, hypothesis here, but, I wouldn't, I don't think that makes it.
You're not contributing that much to the childcare or the resources by the time.
Yeah, we're down at age 80, right? Exactly. So I think the heritability of that is and another interesting comparison to that, I would say, is regarding the heritability of menopause in women, which is also an age-related decline. Menopause age is also correlated with lifespan. So if you get it later, it's better for your longevity, and that is also around 50% heritable. And I would also argue that that's, by the way, in the shadow of selection. So I think both of those things I agree with what you're saying. And yeah, I think even if I do think something interesting is going on around development. I do think that if you do have some caloric restriction around development, around puberty, around sexual maturity. That's what I meant, not development. Around sexual maturity, I do think that kind of primes your body in a particular way to the rest of life. There's a great paper from, I think it's a collaboration between Gladyshev and Peshkin, who look at rapamycin in mice, which were given rapamycin only during sexual maturity. So like only during the puberty years of these mice. And you do see, I don't remember, there's a sex effect here. But I think in males, if I'm not mixing it up, you do see that even if you give rapamycin just during their puberty, it does extend to their lifespan at the end of life. I would also argue that kind of morbidly, you see it in Holocaust survivors. Many Holocaust survivors were around puberty aged when, the Holocaust happened. So they obviously were facing a caloric restriction, to put it mildly. And I would argue that also kind of primes their body in a way for this kind of slower aging. But again, I would argue that slower aging is by a few percent at most. It's not 20% like you see in mice. So there is some plasticity in humans. It's just a lot narrower, would be my argument.
Right, which falls within the evolutionary prediction that it should reflect where the contribution is. And if that caloric restriction, if living 5 more years would not give you another reproductive cycle, because living 5 more years in your 80s doesn't give you that, then we should have this very sort of narrow impact on reproductive probability. It leads us ultimately to the question of, if we assume that our colleagues who are claiming that we're going to someday live way past 120 were to be the case, if this were ultimately where humanity's headed, maybe not in 5 years or 10 years, as some people seem to be claiming, we're certainly nowhere near escape velocity, but if it were to happen decades from now or a century or two from now, if your hypothesis that it is roughly half in the genome, how would you, if you were deciding how to spend $1 billion on the next true longevity intervention, how would you spend it?
Yeah, so that's obviously the multi-billion-dollar question. I would argue, I don't think I've maybe some people say it this way, but I haven't heard it put this way. In my opinion, in the radical life extension longevity field, there are 2 different camps. There's the camp that says we can reverse aging, and there's the camp that says we can slow down aging. And to me, those are fundamentally different things. Okay, those are completely different things, and there's no easy mapping between them. So the epigenetic camp So the epigenetics say that we can reverse aging. So if we think again of this ball trapped in a potential well, we're saying your well is flattened. I'm going to push it back. I'm going to push back your well. You know, when you're almost in a plane, when you barely have any, restoring homeostasis, we're going to restore that homeostasis. And the people who slow down aging say, well, I don't know how to do that, but what I can try and do is to make sure that your valley flattens slower, which is slowing down aging, but not reversing what's been done. Personally, if one of them were to succeed, I definitely more believe in slowing down aging rather than reversing aging. To me, intuitively, that makes a lot more sense. If we think about it in terms of physics and entropy, like even if you repair, you're trying to repair the most complex machine available out of 10 to the 15, cells in a body, there's a million ways to die. And you're trying to somehow return homeostasis, not to a particular function of a particular type of cell, rather to this whole organism. I'm skeptical. It may happen. Again, I don't know. And there have been lots of scientists over the years who were, whose predictions were overturned. So I obviously don't know. And in my opinion, nobody knows. So anyone who says he or she knows, in my opinion, is not telling the truth. Maybe they think, but they don't know. No one knows. So my hunch is to say that I am more skeptical towards the reversal of aging. So I believe more in the slowing down of aging. Now, how do we do that? That's a good question. So we have lots of hints also from the animal kingdom. There were 2 nice papers, I think, this year or last year, Nature and Science, one on the bowhead whale.
Dennis Fearsanoff. Yep.
Yeah, and also on the naked mole rats. And what you find in both is a really robust double-stranded break repair mechanism, really good DNA repair, double-stranded break repair, which is important, not single mutation.
Yes, it's entirely maintenance of youth. It is not reversal of aging. It's exactly the distinction that you're making.
They seem to have this very good maintenance. So in our model, let's say if we were to have much better enhanced, double-stranded break repair, in the model that we're talking about, and in the picture of a ball in a potential well, it means that the well would flatten slower. So I think that's interesting. Removal of, senolytics, removal of kind of damaged cells, of these cells causing inflammaging, maybe that also could work. I don't know. It's not something that I'm working on myself. Like I said, I'm more on the modeling physics type of. But again, like you put it, I personally, intuitively, don't believe in full body rejuvenation. I think if we were to see something that were to extend maximal human lifespan, I do believe that it would be more about slowing down aging than reversing aging.
Yeah, you can kind of picture a shop with tens of millions of aisles full of tens of millions of glass artifacts, and you either have the strategy of not letting small children run through the shop and knock things over, or you have the strategy of letting small children run through the shop and knock things over, and then claiming that you can do some kind of single intervention or stack of interventions that can undo all of the shattered glass artifacts on millions and millions of aisles. And the problem with that kind of stochastically distributed damage is that the more pockets of damage in which the damage is non-identical, the more impossible it becomes to do a limited finite set of interventions to restore that. There's not an identical artifact on any aisle. There's not an identical artifact that hits the ground and shatters in the same way. This kind of scales to how a multi-trillion celled organism like ourselves, in which each cell has slightly different organelle damage, slightly different accumulation of toxic metabolites, slightly different disruption of chromatin packaging, slightly different inherited somatic mutations. All of these things, these acquired different somatic mutations in different tissues, all of these different things combine with different levels of extracellular matrix damage in each separate tissue. And we don't have any realistic mechanism. And I think it's super important that people within the field keep on pushing back against people who keep claiming that all we got to do is dial back the epigenetic, the epigenome to the settings that it was on 10 years ago, 20 years ago, 30 years ago, because that's great if you can get your epigenome looking like when you're 25, but it doesn't take out the damage that you got in the half century since you were 25. And that distinction is something that I think we have to keep on pushing back on, because it constantly gets blurred. And in some cases, some of our colleagues have actually just on the face said, reversal of aging is something that we're either doing or we're about to do. And it's not even something that has realistically been proposed at this stage.
No, absolutely not. I think people who are claiming that are probably claiming it based on studies on, the aging markers of a particular type of cell in an isolated part of the body of a model organism. And that's amazing. Like, the science is incredible. I'm not in any way denigrating the science. I think it's different though from like what you said, this whole body organismal aging. So yeah, I think those things are just two different things. And we don't have, in my opinion, evidence that we can reverse back the clock. I don't think so. Yeah. So I think what you said is kind of, this is something that, let's say, the reflections of a physicist coming from a non-applicable field of physics, astrophysics, where, there's no money involved because we're just doing it for the hell of it. And suddenly you come to this field where there's lots of money involved, and every conference you go to, I don't know, half or, a quarter of talks are from industry. So it's definitely a different, it's a different environment in the sense that suddenly you feel like people are trying to sell stuff. People are trying to prove something, especially when it's industry. And in that sense, it is kind of, it is what it is. Of course, I understand. I mean, the money pouring into longevity is only getting bigger and bigger. But as like a pure scientist here, who, like I said, I don't have any skin in the game, I sometimes do wish we were a bit more candid about what we know and what we don't know, and what's true and what we think is true, or what we don't know is true. And there's a lot of hype in longevity, and a lot of, it's also, this is kind of me ranting about the field, but you go to conferences and, half of the, like I said, I don't know, half of the talks are industry talks, maybe less. And industry talks, I mean, that's also a disorienting experience for me coming from a completely non-applicable field, because industry talks are like, they share none of the values of an academic science talk. In an academic science talk, it's all about transparency. It's all about, here's exactly what I did, so you can do it too and see that I'm right. So we can see if this is for real. And in industry talk, it's all about, look at these amazing results we got. I can't tell you how we did it. And you're just sitting there. So why are you telling me? Like, why, I mean, if I can't understand the method, why bother? So it's definitely sometimes frustrating as someone, like I said, coming from a more pure academic background in the field, but that's fine. I mean, that's the nature of the field, and money pouring in also means going money for research, which is good. But yeah, I think the field would definitely benefit from less hype and a bit more rigor. And probably that's not going to happen, because money is, more and more money's pouring in, and that's what money does.
I think that the observations that my colleagues who are coming from the physics side are contributing are incredibly valuable. We really, really, really try to amplify the voices of scientists like yourself, scientists like Dr. Fedishev, who have the training in physics, because a lot of times the biology world inevitably enjoys this space where people almost feel like they're living in a pocket of science that has escaped from physics, and it hasn't. Life fundamentally is an entropy amplifier, and the way that we get these wonderfully organized systems isn't by escaping the physics. It is by amplifying the physics. It's the reason why life works as a dissipative adaptive system, because we're entropy multipliers. And it is really, really exciting to see the work that scientists like you are doing to actually kind of put the brakes on the sales and the hype to clarify why we've made these lifespan gains in the past couple of centuries and to define what the actual problems are. So if we're going to claim that we're going to solve the lifespan limit for humans, these are the steps that we actually have to take. So thank you for taking the time to clarify this for us, to outline what the work says, what it doesn't say. If you can wrap us up with your thoughts for the person listening who's interested for their own longevity, what should their takeaway be?
Yeah. So, I mean, I would say that, both our work, my work on the limits of maximum lifespan and on how much our genes impact our lifespan, at face value could be taken in a way that takes away agency. And I understand that, but that's not the message that I would want people to go home with. I would say that, 50%, which isn't genetic, we still don't know what it is. We talked about stochasticity, maybe lifestyle, but still, like we talked about, I think believing you have agency is healthy. Healthy mentally and physically. Living a healthy lifestyle is not just about adding on, X more months of your life. It's about how well you live your life and how satisfied you are of your life. If you're feeling energized and vital and enthusiastic. And I think all of the stuff that are ultimately good for lifestyle and longevity, that are good for longevity extension, not maximal, but life expectancy, eating well, exercising, those things are also good for well-being. And I think it's not a coincidence that it's correlated, that what's usually correlated, what's good for well-being is also good for longevity. So I think my message would be, still live healthy, eat well, exercise, spend time with your loved ones. And it's a lot more about how well we live our lives rather than reaching, a particular high score of living to a particular age.
Awesome. Thank you so much for taking the time to come on Saving Sapiens and explain where the cutting edge of the research is. We appreciate it.
Great. Thanks so much for having me.